Okay, so let's go over exam 2. I just downloaded a fresh one. Where did it go? Oh, okay. Oh, nope, that's not it. Okay, that one's already filled in. Okay, so let's go through this one together. Okay, so here, short answer question 1. I want to figure out when I would start translation. So here I have the DNA sequence starting at the +1 nucleotide, so I know that that's not actually where my start codon is. I'm going to start at the first AUG, right? Remember, that's my start codon. The finding can also be in other places, but it always starts it. So I want to scan this until I find my first AUG, which is right here, okay? This down. Okay, to the tRNA in the figure, add the anticodon to the correct location and the amino acid. So my first tRNA is going to be methionine attached, and it's going to have the anticodon that goes with AUG, so it's going to be UAC. Okay, what are the first 3 amino acids of the polypeptide that will be made from the RNA? So after that AUG, I have a GGC. So let's look up GGC. Because right here is a glycine. GLY. Actually, let's add the MET first. GLY, and then it's going to be UAU. So UAU, it should be a tyrosine. TYR. Okay. So up here, let's just put it here for easy access. Met, gly, and tyrosine. Okay. When will translation end? At the stop codon. Remember, the last amino acid is actually the one right before the stop codon. That's added right before the stop codon. Okay, that the stop codon doesn't actually have an amino acid attached. It's just a stop. And that's why it all falls apart, is because there's no amino acid that is added. Okay, the DNA sequence below is found in the middle of a gene and is transcribed from left to right. Okay, direction of transcription. So the first thing that I want to do is actually figure out the template in the coding strands, which is what it wants me to do anyway. So I want to read DNA 3' to 5', okay? So I need to read it, since I'm going left to right, I need 3' to 5', right? So this top one is going to be my template, and the bottom is going to be the coding. Transcribe the sequence into RNA and include the 3' and 5' ends. Okay, so it's going to be the same as what my coding would be, except for the U's. So 5' A, U, C, U, A, U, G, C, C, U, A, 3'. What are codons and how are codons involved in translation? So codons are 3 nucleotides that match or complement, actually, are complemented by anticodons on tRNAs and specify AA to add, so amino acid to add. I accepted a few different answers here. The main thing here was that it specified the amino acid to add to the growing polypeptide chain. What 3 RNAs will be involved in translating the sequence into a new protein? So we have the mRNA, which is the message, and it determines AA sequence. The tRNAs bring over the amino acids and transfer them, right? Them to the polypeptide chain. And then we also have rRNAs, right? So all this happens on the ribosome. So that ribosome kind of serves as the base for translation to happen. All right, next we're talking about tigers. So the corin gene is expressed only in hair cells in tigers that make stripes. interesting fact about the corin gene, in some species like us, it can also be found in the heart cells, and other places too. Anyway, okay, so A, add in and label the corin gene below to include the +1 nucleotide, the transcribed region, start and stop codons, the switch, and the coding region. Okay, so that's a lot. So let's first start here and determine my direction of transcription. It's left to right because my promoter is here on the left. My, I'm going to switch colors. My +1 nucleotide is going to be right after the promoter, right? So I've got that. My transcribed region starts at the promoter and ends at the end. We didn't really go over this summer all the stop signal for transcription. There is one that you can learn about later, maybe in genetics or some other time. So we've got the transcribed region, we've got the start codon, we've got the stop codon. The striped switch seems to be like here. And then the coding region is the region that will be translated. So it starts at the start codon and actually ends right here. But I accepted kind of any of that. Coding region is here. It's the region that will be translated. The main thing was that I need to start at the start, including the start codon, and then stop around the stop codon. Okay, which of the following would you find in a tiger's blood cells? So the coding region for the corin gene, yes, part of the DNA. Promoter, yes, DNA. Activator is no. Protein specific to hair cells, only some hair cells, right? And then the striped switch, yes, because it's part of the DNA, right? So the switch is part of the DNA, the promoter is part of the DNA, and the coding region is part of the DNA. And so those are all going to be there in all cells, all somatic cells, and germline cells actually too. Okay, in tigers, the corin gene is expressed only in hair cells that make stripes. Okay, we know that. A mutation in the corin gene is specific to golden and snow white tigers, resulting in berry stripes. This mutation changes the amino acid from a histidine to a tyrosine. Okay, so that sounds like a missense mutation. Okay, the histidine to tyrosine mutation has replaced a positively charged amino acid with an amino acid that is hydrophobic and doesn't like water or charges. Okay, so big change. Big AA change. Okay, so what type of mutation is this? All right, I already said that. It's a missense. Where would you find this mutation in the DNA? Okay, so here I could find it in the coding region of the DNA. Has the primary level of protein structure been changed? Okay, so the primary level of protein structure is just the order of amino acids, right? And that has changed. So yes. So since the order of the amino acids has changed, we have had a change in the primary level of protein structure. Has a tertiary level of protein structure been changed? Probably. I also allowed yes or probably. The AAs are very different. So that would probably affect the way that it's folded. And then has the function of the corin protein been changed? So here you do actually know the answer, right? So that's where you could be like, actually, I'm going to say yes. And that's because I can see a phenotypic change, right? So it must have changed the function, right? And has this protein caused a change in genotype, phenotype, or both? So both. The DNA is changed, and patterning or phenotype is changed as well. Okay. All right, next is to look at the PIDX1 gene. So remember that we had these different fish that either had pelvic spines, and that was good if there's largemouth bass predators, right, around. But it was bad if there's insect predators. So if there's insect predators around, it was better not to have a spine because there's these little like crabby dudes. It's a crabby dude with these claws that would try to catch you, right, by your spine, okay? All right, so the PEDX1 gene encodes for a protein involved in the development of the jaw, the pituitary gland, and that pelvic spine in the stickleback fish. And the picture of the PEDX1 gene labeled the number with the number where a mutation would be located that would result in no expression in the jaw. So no expression in the jaw would be right here. No expression in any region would be in the promoter. A nonsense mutation would only be in the coding region. And then the fish that doesn't have a pelvic spine but is alive, right, would only be in the pelvic switch. Okay, if we had it in if we had it in the promoter or in the coding region, then we would just have a dead fish, right? All right, next was to look at some codons, figure out what the mutant amino acid would be and what would happen to the protein. So here we have the mRNA codon CGA, and that encodes for arginine. Here we have a mutant codon where instead of a C, it's a G in that location. So we have GGA. So GGA is going to be glycine. So it switched from arginine to glycine. That's a missense mutation. And the likely impact is unknown. There's definitely a change in primary, and then maybe a change in tertiary. We don't know. Okay, could be a big deal. It could be not a big deal. UGA. Oh, UGA I think is a stop codon. Yeah, UGA is a stop. Right, this is a nonsense mutation. It's nonsensical, and it breaks the protein. Right, so you could have said early termination. You could have said just that it changed the protein. I accepted a few different things here. And then AGA, so AGA is arginine again, right? So arginine, there's no change in the amino acid that it codes, right? So that's called a silent mutation because we don't even see it in the primary structure. And there's no change for the protein. Okay. The next one, imagine that you are a cardiac doctor looking at the role of this gene MYH7 expression in adult hearts. You took blood samples from two people, one with cardiac disease and one without. You have decided to compare their DNA to see if there's any differences in this gene. Can you use the blood sample to compare the DNA of the two people to see if there could be structural differences that in the protein that's made? So yes, the blood would contain the MYH7 gene, and I could figure out the amino acid sequence made from each sample. And then can you use the blood to compare the amount of MYH7 protein that is made between the two people? So my initial thought for this was no, because the MYH7 gene is not expressed in the blood. But I ended up accepting a few different answers here, actually. One was maybe if there is a difference in the promoter or switch, right? So some of you were like, well, I have the DNA. I can look at the promoter and the switch as well, and not just at the coding region. So I allowed that as well. Next, choose which of the following will be more similar and explain your answer. So all of the proteins found in blood. So all proteins found in blood between two people. So in blood between two people. Or all of the proteins found in the blood and in the heart of the same person. So this one was both in the blood. This one was in two different areas, but in the same person. So remember, when we have proteins, right, we're going to have more similar expression in the same tissue type, right? So expression proteins, right? That's the end of like the proteins are the end of transcription, translation, central dogma, right? So expression is more similar in the same tissues, right? If instead of asking all of the proteins, if it was all of the DNA, then it would be option 2. But this is asking specifically about all of the proteins, right? Okay. Then we kind of move on to ecology. So in the movie The Lion King, there's a conversation between King Mufasa and his son Simba that goes like this. And it says that everything you see exists together in a delicate balance. As king, you need to understand that balance and respect all creatures, from the crawling ant to the leaping antelope. And Simba asks, but Dad, don't we eat the antelope? And Mufasa responds, yes, Simba, but let me explain. When we die, our lion bodies become the grass, and the antelope eat the grass. And so we are all connected in the great circle of life. Okay? So I wanted you guys to think about the pyramid and the food web, right? And think about how there's producers at the bottom, and then multiple levels of consumers as you move up. Okay? So the antelope are going to be primary consumers because they eat the grass, and they're going to be heterotrophs. They don't make their own energy. They use the energy they get from the plants. The lions, you could have either said secondary or tertiary consumers. I accepted both answers there. And again, they're heterotrophs. They're going to be eating the antelope. And then the grass is the producers. So they are going to be autotrophs. They use the nutrients in the soil that have been broken down by the lion bodies, but they use energy from the sun, right, to actually grow and get energy. What category of living things would help break down the bodies of the lions so that their bodies become the grass? So decomposers would do this work. What types of living organisms could play a role in breaking down the bodies? So we could have lots here. So you could have said bacteria. You could have said animals. And fungi, I think, were the 3 that I required. You could have also had protists here, potentially. We could have also had archaea here. But we didn't really talk about those, okay? Can nutrients be continuously recycled? Yes. The decomposers do this. Can energy be continuously recycled? No. We have to, it is lost. You could have said a few different things. It's lost as heat as it moves through the pyramid. So we have to get that continuous input, okay? Next was about the bluebirds. So imagine that you're a homeowner and you have a pair of bluebirds that have made a nest in the same place for 2 years. And they produce healthy chicks by feeding them hundreds of insects, including caterpillars. This year, they've returned, built their nest, and laid their eggs. However, you know that there has been a change in their environment. Your neighbor has a big garden and loves the butterflies that visit the flowers. This year, they have used a pesticide targeted to get rid of the mosquitoes. Without realizing it, they have also killed a lot of the other insects, including caterpillars and butterflies, in their yard as well. So draw a labeled figure and write a short explanation that you would use to help your neighbor understand how their choice has impacted the backyard ecosystem that both of you share. Include the terms population, community, and ecosystems in your explanation. Okay, so here, right, we have you could have done this a few different ways. I accepted a lot of things. There's producers, then we have the insects, and then we have the birds, right? And so the insects are eating the producers, but you don't really care about that part. What you really care about is that the birds are eating the insects, right? And so if you kill all the insects, then the birds also die, right? So you could just kind of write that out in some way. If you kill, and you could have added these in multiple ways. I'm just doing one of them. If you kill the insects, I'm going to add one of those words in. The community of insects, right? So multiple species. Then the population of birds in the area will suffer because they don't have enough food. This harms the entire ecosystem. Right, so one little change that you think isn't going to impact anything, except like whether or not you get bitten over the summer, actually has a huge change in the entire ecosystem. Because if there's no birds, then we don't have other things there as well. Okay, next, in a short article from the National Park Service, they state that under ideal circumstances, eastern forests can sustain themselves without intensive human intervention, but we are currently teetering on the brink of a catastrophic loss of eastern U.S. forests. The main culprits for this impending disaster are overpopulation of white-tailed deer and invasive plants. So if you go out right now to any of the forests, like Daniel Boone, one of the main things that you'll actually see in there is called a bush honeysuckle. So there are native honeysuckles, but there's also this bush honeysuckle that's kind of taking over, okay? And the white-tailed deer won't eat it. Okay? Because they don't like it. The article goes on to describe how deer love to eat native tree seedlings but don't like the invasive plants from other parts of the world. Most invasive plants cannot be used for food by other species either and don't support our eastern forest ecosystems. So like those honeysuckle berries aren't as nutritious as they need to be for our birds, and also there's not as many caterpillars and things on them compared to native species, okay? So use the concepts of keystone species and trophic cascades to illustrate how the loss of deer predators like wolves, coyotes, mountain lions, cougars, bears, and bobcats is partially responsible for why our forests are in jeopardy. Okay, so you could do lots of different things. So you could say, that you have like, oops, where's my thing at? Okay. Oop. Okay, so maybe we want to like try to draw a wolf. Can I draw a wolf? Okay. I don't know why I want it to be fuzzy up there, but I do. Okay, this is a wolf, guys. Has a snout. Okay. Here's a sly little wolf, okay? And here's my deer predator, right? Or my deer. It is my deer predator, okay? So here's my deer. Let me get a boy. Okay. This is a deer. Okay. And it's a white-tailed deer. Shoop. Okay. That does not look like a deer at all. All right. That's all right. So maybe it just needs longer legs. Maybe that will help. Okay. Here's my deer. As long of legs as I can give it. Okay. So here's my deer. And I've got these trees growing, and I've got little baby trees that are native, right? They're trying to grow, and then I've got these bush honeysuckles, right, or whatever. There's other species too that are causing trouble, okay? Okay, and if there were enough of the wolves around, right, to kill off some of the deer, then the deer would stop eating all of these native trees, and they could maybe outcompete some of the invasives, right? But instead, the deer there's no predators, and so there's lots of deer, right? So there's too many deer. Oh, that looks even stranger than it did before. Okay. Let's make sure I can grab all of it. I don't know why it's doing that. Okay, so when there's lots of deer around, right? Then all of these go away, right? The native trees go away, and we have just invasives taking over, okay? So wolves and predators are keystone species, and their loss results in catastrophic trophic cascades that destroy entire ecosystems. Largely because they might keep something else in check, like the deer, okay? All right, so I think that's it for exam number 2. So that's all done, and I hope this review helps you all think about all this content and helps you understand maybe where you might have missed some points and where you can go back and review.